Good day. So this is the second part of our lecture in the microbial world. So we will be talking about the growth of the microbes. So some microbes here are pathogenic. So as we had discussed before, they are the dominant life form on the planet, and there's a very large and they are a very large and diverse group. Now, because of their diversity, some of them are pathogenic, they are harmful to us, some of them are beneficial to us, and some are neither harmful nor beneficial. So for the pathogenic microbes, and even for the microbes that are beneficial to us, especially when we are using them for to manufacture our products, it's important to know how to control the growth of our either our enemies or our friends. So the culturing microbes, or when I say a microbial culture or a culture, it's the cultivation of microbes in an artificial medium containing nutrients. There are various techniques that are used to identify microbes and, of course, to quantify their growth artificially. This is in a culture. So they sometimes called in vitro. So here is a micrograph. This is an image taken under a microscope. This is a gram stain microscope. As you can see, you have violet-ish color. So you have that is because of the gram staining. So the smudges that you see here are the microbes. I think this is the Oreos, stuffed Oreos, or at least they are coccus bacteria. Now, here is what we can see without the aid of a microscope. So this is a Petri dish. So this is a culture of microorganisms. So the white smudges here are your bacterial colonies. And of course, the growth medium is colored reddish. So here is one way to quantify them through the coloration, or rather Beer-Lambert's law, application of the Beer-Lambert's law. Okay, so how do we quantify them? So we can quantify them under a microscope. So this is cell counting. So we have your bacterial suspension. So we put them in a special slide. So this is different from the slide that you can see in the laboratory. So this is we call it the Petroff-Hauser counting chamber. So inside this slide, it contains small grid lines. So you count, basically it depends on the system, but the most typical one is you count the microbes in the square. So you have here a 9 by 9, 3 by 3 or a 5 by 5 squares. But the usual one is 3 by 3. And then you count it on the corners and the middle. You count the squares, the microbes in the squares of in the corner squares and the middle square. So when I say corner square and middle square, here is the one square. So I think this is the middle square. And then you just count all the microbes inside. So the issue here is, or rather one of the challenges here is counting them when they are in the lines. So do you consider the organisms in the line straddling the actual, they are actually or literally straddling the fence? You count them or not? So the thing here is to be consistent, but the challenge is this, for example, is this microorganism part of this square or this square, because they are basically on the line. So when I was working in the industry and I was doing actually this cell counting, I tend to just since for the adjacent squares, I allocate them lines. For example, the L-shaped line here, all of the microbes on this line and this line is counted for this square, but the ones on this line and this one is not counted for that square. So that's how I do it. Basically, this is how you count it, or just be consistent. So that is the cell counting method for microbes. And then the next one, we have your because the use of spectrophotometer. So this is a more this one gives us a more I cannot say accurate, but each of them has their own advantages and disadvantages. For this one, this is not dependent on the person or the human error. Of course, you count them. There's an error in counting. So there is a less human error when you use this spectrophotometer machine. So the thing here is it's based on the turbidity. Because when a microorganism grows in a liquid medium, or rather is in a suspension, they become turbid. So you are just going to measure the turbidity. Okay, so how the light, the OD, optical density, we call it OD 600. So optical density at a certain wavelength. So it can be 600 if I remember correctly, but depending on the specifics of your laboratory protocol, you might use. So you have this turbidity test. So the one of the upside here is, again, there is less chance of um, wrong counting or double counting or even human error, because the Petroff-Hauser counting chamber relies on manual counting. So you have your uh, counting errors here. Now, for this turbidimetry test, it relies on a machine, so there's a less of a human error, but the turbidity can be affected not by the cells, but also from debris, from not live cells, not whole cells, but also from the lysed ones. So there is that possible source of error for turbidity, for the use of OD or the spectrophotometer, as opposed to this one. But for the Petroff-Hauser counting chamber, you cannot know which colony is, or rather, which organism, which of them is actually alive, because of course they're not moving. But you can see the intact cells. Dead or alive, they are the intact cells. Now, the last one here is the more modern, or at least for the three of them, this is the most modern. So you have your cell sorter and cell counter. So you have your mixture, labeled, or it's either labeled and unlabeled cells, and they are moved through a small capillary tube where they are segregated. So you have a laser that can actually count the cells as they pass through, and then you can actually sort them in terms of population. So this is a more sophisticated equipment. And of course, unlike the turbidimeter, which can be affected by, of course, the not only the cells in the solution, but also in the debris in the solution. If the culture is full of debris that are not non-cellular debris, so it can affect the turbidity. But for this one, it only counts the cells itself. So you have a more sophisticated counting here. So these are some of the ways which we can monitor the growth of the microbes, of course, by counting them, counting their population. So aside from, of course, why do we need to monitor the growth? Is because we need to control the growth. So altering ideal growth conditions can control the growth of microbes. So for example, we refrigerate, we use refrigeration. So we refrigerate our food, we refrigerate the raw meats, raw fishes to prevent the microbial growth. Because of course, at low temperatures, the growth is suppressed. So this can also apply to toxic chemicals. So we can also use toxic chemicals such as antiseptics, which is on the living tissues, and disinfectants on inanimate objects. So antiseptics, examples of that is, of course, the alcohol, which we use now during the pandemic, a very ubiquitous facet now. When we go out, there's always, before you enter an establishment, you have to you have to apply alcohol to your hands. And then you also have disinfectants like bleach. So aside from these, we also have the antimicrobial drugs. Well, basically, these are the antibiotics. So antimicrobial drugs, since we intake them, they have selective toxicity. They can kill invading microbes without damaging the host. Otherwise, it becomes just a simple poison. Okay, so they target features unique to pathogens. So what are those features? So it's easier to do with bacteria because they are prokaryotic. So one good target here is the component of their cell membrane. So that's one of the most common mode of action of your antibiotics. So the most common target are the proteins, or even antibiotics that induces holes in their membranes or in their cell walls, because their cell wall is inherently different from us. Now, eukaryotic pathogens are more difficult of a target because they are eukaryotic. Now, for some of the differences aside from the cell walls, one of the more promising things they are looking to is to actually target the ribosomes. The ribosomes of prokaryote is different from the ribosomes of the eukaryote. the ribosomes of the prokaryote, we call it, they are rather smaller compared to the eukaryotic ribosome. So it's good it's actually an ideal target. Now, viruses are also very difficult to target because they are intracellular pathogens. They are very difficult because they enter the host cell itself. And so when it enters the host cell, we cannot use antibiotics there because antibiotics should only work outside of our own cells. If it penetrated inside our cells, it can kill us as well. So for the viruses, they are parasitic. They are cellular parasites. So they hijack the metabolism, the mechanism of the cells, so that the cell will produce the virus instead of actually doing its actual work. So that's the mode of action for the virus. And that's why they are very difficult to target, because you have to kill your own cell. Okay, antibiosis is the interaction between two organisms that harms one of them. So example here is the use of penicillin, the use of penicillin by the penicillium fungi. So most of our antibiotics are actually derived from the antibiosis, the antagonism between the two organisms. So here is an example of your culture of Staphylococcus aureus. Here shown is as a smudge, and then this is your Penicillium notatum. So that's the original that is the organism where penicillin was first discovered. So as you can see here, there is no Staph aureus growing in this area because the Penicillium notatum produces penicillin and it kills any microbes that comes in contact with it. So here is the zone of inhibition where the penicillin actually diffuses to the agar media. So that is your antibiosis. So your Penicillium notatum produces a poison or a toxic chemical that prevents the growth of the Staph aureus. So this is just one example, but there are many different types of antibiotics that are now available. So aside from the health perspective or the medicinal perspective, we also have environmental microbes. So the microbes are the recyclers of the planets. They're the decomposers. They decompose dead organic material. They're also involved in biogeochemical cycles that can convert unusable forms of carbon, nitrogen, and sulfur into forms that we can actually use. Because, for example, in the case of the nitrogen, we cannot use N2 because it's a very, very inert gas. Now, we can only use nitrogen in the form of nitrates or even ammonia or urea. So we need to convert the nitrogen first, the nitrogen gas first into the usable form. So for the plants, microbes and plants can form mutually beneficial relationship. So example of them are the rhizobium and the legumes. So rhizobiums are actually these are the microorganisms that actually inhabit the nodules, the roots of the plants. And they are good, they are beneficial to the plant because they fix the nitrogen. They're actually nitrogen fixing bacteria. So they provide nitrogen to the plants. Actually, this is actually the best form of interaction between organisms. Rather than attacking one another, it's better to actually help one another to actually live. So the plant provides nutrients and even habitats for the microbes, while the microbes will provide the plants with their usable form of nitrogen. So many fungi can help plant root systems acquire water and nutrients. So we have here some, they actually, because well, basically the growth of the fungi in the plant root systems. And then aside from that, for the human's perspective, we have microbial remediation, which is the use of microbes to clean the environment. So example here is, of course, I think we had already discussed, or you will be discussing them in biosafety and environmental health. So you have all-eating bacteria that is used to clean the Gulf of Mexico after Deepwater Horizon spill. So example of that. And then we also have the Guimaras oil spill for us. So one of the ways to actually remove the toxic oil or toxic sludge is to have the bacteria eat them, degrade them. So aside from the environmental uses, you also have in the industry, we have bioreactors that are used to grow large amounts of bacteria with special properties, such as the industrial production of vitamins, alcohols, and other pharmaceutical products, including we have here some of your insulin, some of the drugs that we use. Okay, so those are industrial, that is, here is an example of a small lab scale bioreactor. Okay, so in the food business, or in the realm of the culinary arts, we have, of course, the fermented foods. We have the yeast; it's used in production of the beer, wine, and bread, and of course, the bacterial culture for the production of yogurt, cheese, sauerkraut, pickles, and then for us in the Philippines, we have the bagoong, patis, what else? Toyo. Okay, so you have those, or buro. Buro makes use of the microorganisms inherent in the plant skins, to actually ferment the food. So you have example is burong mangga, burong talangka, so something like that. Okay, so in also in the food business, the microbes here can be both beneficial and harmful. So they become harmful because they are the cause of spoilage. So that's why we have pasteurization. It's used to kill. It uses heat to kill microbes involved in food spoilage. So almost all, no, all of the manufacturing business should be doing pasteurization of their products, so as to prevent microbial growth, especially in the dairy products. So you need to have pasteurization here. So lack of pasteurization is linked to disease because the raw milk, when it's squeezed off from the udder of the cows and goats, you have it actually also contains the microbes, harmful microbes, salmonella, something like that. So they are linked to disease. So they must be first decontaminated by sterilization. Okay, so these are some diseases caused by contamination of unpasteurized milk. So you have here Brucella abortus, which is related to brucellosis. You have gastroenteritis, which is caused by Campylobacter jejuni. So these are just some of those examples. Okay, so these are some documented outbreaks associated with unpasteurized milk in the US. So this data is from 1993 to 2006. So as you can see here, the outbreaks, the bar graph is actually linked to the spikes of the illnesses. Here is a bit of anomaly, of course, even because there is a large outbreak, but there is just a small amount of illness. So probably it was prevented or probably not much registered. So all in all, as a summary of our lectures, the first part as well and this part of the lectures, so microbes are the most abundant and diverse life forms. They are involved in complex interactions with humans, whether good or bad. They can be the cause of infectious diseases, but they can also be used for human benefit in the industry, the food, and environmental applications. So that's it for today. So copyright this lecture slide is copyrighted to John Wiley and Sons Inc. of 2019. So thank you for listening.